All right, so I think we can get started. After a quite handful of methods that you used yesterday, like MNN, also CCA, the goal of this lecture now is also to give a background on other dimensionality reduction methods, and probably also give you a little bit of understanding how they work in the essence. I can also say that I'm not going much into details how exactly those methods works, and some of the steps into how they really work, let's say math formulas and so on, I'll just skip it. What I want you to get from today is the intuition, how they work, and how you can use that information to tweak the parameters and see how visualize your data or reduce the dimensions of your data using PCA, tSNE, and UMAP. So my name is Paulo. I'm part of the Elixir Sweden, called NBIS, National Bioinformatics Infrastructure in Sweden. And today we're talking about 3 different methods. So dimensionality, why do we need dimensionality reduction? So far we have been working, for example, we have your reads, your counts, and these are what you already get from a sequencing facility in general, until you have your count QC that you have worked yesterday a little bit. And then you normalize your data, as you've seen that as well yesterday. But then you go for a dimensionality reduction technique. Either it could be PCA or CCA, depending on which steps you need to do, if you need to integrate your data and merge them. But in general, you go for PCA, and then tSNE or UMAP, that I'll explain today. And then go that for clustering, and then differential expression. You do it for the clustering to identify your cell types if you don't know them beforehand. But of course, for a single cell strategy, there are many options that you can choose. So you can go directly from normalization to differential expression if you already know your classes. So then you actually don't need to reduce the dimension that much. And then you can go even for trajectory analysis, and there are many other options that you can do afterwards. But there are many other paths. So you can, from PCA, go directly to clustering, from normalization directly to tSNE. And that is really data set dependent. And I'm not going through every single detail why you should do each one of those. But the workflow, the arrows here would be like the standard way of doing analysis, let's say. And the workflow will depend much on research questions. If you have batches, then you use CCA or NMM that you saw yesterday, or yeah, many other factors. So why do you have to do the dimensionality reduction? So one of the reasons is to simplify complexity, so it becomes easier to work with. So you have a matrix with thousands of genes, and then you reduce it to 2 dimensions. And in some cases also transform non-linear relationships to linear, so it's much easier to work with them. So especially for clustering, that will be a big step that you'll see today also. So another thing is remove redundancies in the data, because many genes are quite correlated to each other. So actually we don't need thousands of genes to explain that two samples are quite different. We can reduce the dimension, and I'll see that in PCA how that works. And also identify the most relevant information in your data. That's also what PCA will do, and I'll talk about it. And also reduce computational time, because in many cases you can reduce the dimensions of your data to a couple of data points. Instead of using 20,000 genes, I can use 20 principal components, and that is representative of your whole data. And I can use only those part of the data to continue, for example, clustering, differential expression, or so on, or yeah. So then that also facilitates clustering. And of course, as you might know, visualization. That, for example, you've seen UMAP plots yesterday, or this is also another one, the tSNE. So dimensionality reduction algorithms can be divided in 2 or 3 groups, depending on the nomenclature. So here I'll just put in a list of all of them. At least, there are, of course, many more. So they are divided in matrix factorization. So you have a multiplication of matrices, and by using that method, you can reduce the dimensions of your data. So here you have PCA, ICA, for example, and MDS. Maybe you have heard of those 3. These are more advanced versions of those here, accounting for like the sparse data that single cell is, or yeah. And there's these graph method-based algorithms. So we have tSNE here, UMAP, large Vs. These are the most common ones. Diffusion maps I'll talk more about during the trajectory analysis. And there is also the new version of it, which is autoencoders. And those autoencoders are also kind of could be inside the matrix factorization because they are based on neural networks. But today we're talking about PCA, tSNE, and UMAP. So let's go for the first one. PCA, how many of you are already familiar with PCA? Raise your hands. Yes, so quite a good part of you. So then I can also go just through the intuitions of it. So it's a linear algebraic method for calculate dimensionality reduction. It's a case within single value decomposition method that used for data compression. And it says that any matrix can be decomposed as a multiplication of other matrices. In other words, this is eigenvalue decomposition. That's one of the tricks of PCA. So let's visualize that in a little bit. So imagine that you have the original data as the matrix M, and then that can be decomposed as the multiplication of these other matrices. I'm not going much in details exactly on which one, but what we need to know is that you have some samples in here, some genes in here, and that's basically every matrix in biology, let's say, that we use for RNA-seq or single cell expression. That can be decomposed in the principal components and the genes. And these matrix here will give you the importance of every gene to each principal component. And principal components will be the reduced space that you're getting into. And I'll explain in the next slide how you get to this point. And the other matrix is your samples to the principal component will give you basically the visualization that you want, separating your samples based on the principal components. And this matrix is also not present during PCA because usually we scale the data beforehand. And one of the steps you did yesterday was scale the data. And I'll go a little bit later on that during the exercises. So this matrix is kind of absent for PCA, for the standard PCA. So it's the multiplication of those two matrices. If you go for a principal component object, this is what you get. So there are two matrices here, rotation and the X. And those correspond to these exactly two matrices here. And the center is scale. If you run standard PCA, will be basically empty, those two vectors. So how does PCA work? So if you have a PCA, the two genes, let's say gene expression A and B, the first thing is center the two genes. So the mean of them will be 0 here. So they are kind of comparable. It's not that you have one higher than the other. After that, you pass a line through the whole data. If you have many genes, then you pass a line through all the genes in one go. But here you can see a line in red that expands most of the data in this axis here. And a blue line that is 90 degrees to the red line. So the red line explains the most of the variation because the dots are spread more over the red line in this axis here than in here, right? So if you link each point to the blue line here or in the red, so this is the distance of every point in the red line. This will correspond to principal component 1. If you do on the other way around, all the distance from each point to the blue one, in this blue axis here, that will be principal component 2. So this is how the PCA of this data set will look like. So then you have this, most of the points spread over principal component 1. And some points spread over principal component 2. And you can see that this matrix, the PCA, is basically just a rotation of your data to another coordinate system. So you're basically rotating the data so that principal component 1 will correspond to the highest variance in your data. So it's just a transformation of your data to that coordinate system. And as you can see here, the red lines here in the principal component 1, they are quite much bigger than the ones in the principal component 2. That also indicates that principal component 1 stores more variation of your data. And you can see as the variation of those dots will be corresponded in here in this plot. So 98% of the variation in your data is represented in principal component 1, as opposed to maybe 2% in principal component 2. And because of that, PCA can then, PC1 explains 98% of the variation. And principal component 1 then represent those two genes well, because they are highly correlated in this example. So then you don't need two genes to explain this phenotype. You just need one principal component. So let's say that you have 20,000 genes. Principal component 1 will actually represent maybe half of those genes very, very well. That depends also on the data set you have. And that's how you can also remove redundancy in your data. And principal component 2, in this case, is nearly insignificant because the amount of information stored in here is quite low. And then we can just disregard this one and continue our analysis on principal component 1, if this is the example. Now, if you have 20,000 genes, maybe you'd select maybe 10 important principal components and so on. But in real life, this is how it looks like. So there is not a clear cut as opposed to this one. So then you can cut your data maybe here, or maybe use the top principal components that are in here. I think it's 50. We can't see because of this. But so this is what you're actually doing later in the exercises. Selecting the top principal components. And that's what it means. Get the most information out of your data. In single cell data analysis, you can sometimes also differentiate some cells based on the principal components. Some of the gene patterns between your cells will be captured by principal components. So these are two different data sets. But sometimes these principal components also would capture the sequencing depth of if you have two different libraries. So they will be separated based on the sequencing depth, or also by heterogeneity and complexity, especially if you have red blood cells, because they are very small and they don't have that much RNA compared to, let's say, fibroblasts or epithelial cells. So then they will be separated not only by the expression pattern, but because they also have completely different quantities of RNA quantified in our data. But is that not always the case? So it really depends on the data set. And looking at one of the matrices that I mentioned before, so it's principal components versus samples and principal components versus genes, that those two matrices give your original matrix. And because of that other matrix, we can identify what is the importance of every gene to every principal component, how much those genes will be correlated to principal component 1 or 2. So here we can see, for example, IL-32 and this other gene here, they impact a lot on the principal component 1. And these other genes also will impact a lot, but negatively. So they are inversely correlated to principal component 1. And this is another plot that you can usually see if you're using the default pseudo-out method. And then you can see which genes really correspond to some cell, separating some cells in principal component 1. But we will go through this a little bit on the exercises. But that's the idea that I want to give it to you. Some genes are more important to principal component 1 or 2. So in summary, principal component is a linear method for dimensionality reduction. Each principal component is interpretable. So you know that principal component 1 has more variation or less variation. And they mean something. So maybe some genes impact on that principal component compared to principal component 2 that other genes contribute most. So the data is usually scaled. So all the expression of every single gene should be 0, and the standard deviation should be scaled to z-score normalization. And that's here, z-score. You can also do that by hand. Or if you're using pseudo-out package, there is a function scale data. And that's why you do that function before computing PCA. And you can also select the top principal components because they store highest variation. And therefore, you can filter your data based on that, either using, for example, the human-defined threshold, all the principal components that explain at least 1% of the variation, or a Jackstraw, which is a method that is implemented in pseudo-out. But I'm not going into details, but please check this out. And also maybe use the first principal components, let's say 5, 10, or even more, depending on your data set. But it has also some problems, since it performs poorly for single cell data sets to use as a dimensionality reduction and visualization method, because it's single cell has 0 inflated data types, or like there are many zeros, and that impacts your PCA a lot. And the cell sizes and sequencing also are captured by principal component, and usually that's not what you're looking for when looking at single cell data. And that's why you would opt to use another method that is non-linear transformations, or dimensionality reduction. But before going to that, yesterday you listened a little bit about graphs. Today I'll just want to give a brief introduction to graphs, because that's where tSNE and UMAP are based on. And later today you'll hear a little bit more even about graphs. So this is a plot, and this is a graph, because in some languages, they mean exactly the same thing. Let's say I'm Brazilian and Portuguese, graph and plot is exactly the same word. So this is what I mean when we mention about graphs. So each dot is a cell, or it can be also a gene if you transpose your matrix. And each line represents a connection between those two cells, saying that they are strongly correlated, for example, or they are very close in a multidimensional space. So it's a easy representation of your data in this way. And each connection can be weighted as a proximity of cells, let's say correlation or Euclidean distance. And there are many other ways of computing or giving weights to these lines here. So every nonlinear or graph-based dimensionality reduction algorithm is based on this concept. So the first thing you do is there are two steps. You construct the weighted graph, just like this one, based on the k top connections. Let's say the top nearest neighbors. You probably heard a little bit of that yesterday. And that's also called that k and n. And then you also do a low dimensional embedding of your data to this graph. So for example, this graph above, you have, I think 13 points. So you'd have a matrix with 13 by 13. And when you do this graph layout, you reduce the dimension to 2 dimensions, right? Because you have like the graph here and here. So this is what we call an embedding. So transform your distance matrix of 13 by 13 to a 2-dimensional space, linking these are closer together, these are closer together, and these are closer together. So what tSNE and UMAP will do is find this plot here that matches better with the original data. So it's an optimization of this graph here, because there are many algorithms to generate it, and tSNE and UMAP try to optimize that part. So let's go for tSNE. tSNE stands for T-distributed stochastic neighborhood embedding. And I'll explain during the presentation what these exactly mean. So tSNE is a graph-based nonlinear dimensionality reduction. And it's, I mean, the data in a multidimensional space is not linear, as I've been mentioning a lot. And this is one example. If you have a manifold or a data that is structured in a way that is not a plane, let's say, in here you have a spiral of every point here, it would be, for example, a sample. And here you have this dot in white and this dot in white. So if you just compute a PCA and try to calculate the distance between those 2 points, this will be the distance, a straight line between them. In what tSNE tries to do is to find the distance between those 2 dots, but now considering all the neighbors that it has close to them, or that the neighbors that can link them together. So actually, there are no cells in this space. So the nearest neighbors will be always here, 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 all the way there. So this is the distance that you get in the tSNE space, let's say. In general terms, of course, because there are many parameters to change that. Yeah, so tSNE will calculate the distance based on the weight of the neighbor's cell. So how does it do that? How do you make sure that the space that you reduce your dimension to is equal to the multidimensional space of 20,000 genes that you have? So one of the ways to do that, so for example, imagine that you have this multidimensional space and you have different samples here, and you want this layout to be quite similar, also represented in a low dimensional space. So it uses, for example, a t-distribution around this center here to estimate the distance. So the furthest points will not even be considered because the distance will be close to 0 in that t-distribution. So for example, a point that, so imagine that this is the point I that is in here. And a point that it will be in here will have kind of this value as closeness to it, rather than the ones that are very far apart, like these ones, will basically have a 0 as a distance. They will be not considered at all. So this distance between point I and J will be considered as the probability of J be picked as the closest point to I. And this same probability should be the same probability in the reduced space. So the probability of if you is my nearest neighbor in the multidimensional space, in the reduced space, you still need to be my nearest neighbor in the reduced space. Yeah, so this is just in general terms. The probability of those two in the multidimensional or the low dimensional space should be very similar to each other. And that's what, yes, please. Correct. So the idea is that when you optimize the graph, this graph should really represent your original data in terms of data structure, let's say. Yes. That depends. So by default, tSNE can use Euclidean distance just because it's calculated. So it's a linear distance between this cell and the nearest ones, but not across all of them. But you can also use any other distance matrix. You can actually input the distance matrix as input for the tSNE if you need. Yeah, but as default is usually Euclidean distance. So let's try to visualize it in a more didactic way. So imagine that you have gene A and B, an expression, and let's say that you have 3 different clusters, the red, the blue, and the green one here. And you want to embed this into a one-dimensional space, so reducing the dimension. So the first part is a stochastic embedding, and that's what comes from the name. So t-distributed was the previous part, how you define the radius of your nearest neighbors. And now the stochastic part is this one, that you embed your cells stochastically, just at random. So you put all your samples here in a one-dimensional line, and they are distributed randomly. And that, if you mention, for example, measure the distance between the red cells here and the red cells in here, you see that they are quite different. This is quite big compared to the original space, right? So this is a bad embedding, let's say, of your bad representation of your data here. So by the means of those distances that I was mentioning in the previous slide, those distances is actually mean as Kullback-Leibler divergence. So it has a higher error, let's say, in your representation. So that makes sense that some of those cells we need actually to push them together in here, so that those 3 cells become quite closer together as in the original space. So that gives a weight to these cells to move up apart where they are and move together. But we also have some cells, for example, these green ones and the blue ones here, they are not together here, but they are together close to the red ones. So they should be pulled apart from the red ones. So we can do the same thing from the blue ones. You attract the blue ones all together because they are actually close in the multidimensional space. And the other ones will try to be repulsed. And then you do that several times. So now you update the new positions of those cells. And then the red ones are still getting closer. The blue ones are starting getting closer. The green ones as well. And then you keep iterating over time until you start converging. So that's the idea of tSNE in very simple example, let's say, that now you have an embedding that has the distance between your red cells here are very close to your multidimensional space. And as for tSNE, it does not really concern about the distance between the furthest apart cells. So for example, the distance between the red and the blue and the green is not that important for the tSNE. If you have, for example, those 2 clusters swapped, even for the tSNE, you will not consider this as really problematic, as long as they are far apart from the red ones. And in this setting, this is how you have like a lower divergence between the reduced space and your multidimensional space, because the distance in here is close to the distance in here. As well for the blue ones, they are close in here, and the green ones, they are close in here and in here. And you cannot read this one. So the same concept applies to any other dimension. So you have 20,000 genes or maybe 30 principal components, and you reduce that to 2-dimensional space using this concept, let's say. However, there are many hyperparameters, or what does that mean? It's like tuning factors that you can alter, for example, to define your graph. Another method of using tSNE is called also the Barnes-Hoots implementation of tSNE, but developed by the same author. And that actually speeds a lot the computation and is one of the defaults so far. But there's a new one even that I'll mention soon. In this one actually comes in different names. So there is the R tSNE package in R. There's the Seurat package that implements this one and another one. And there's, if you have worked with flow cytometry data, you've heard of probably tSNE, which is exactly the same one here. And these tuning parameters are, there are several ones. There are actually 10 hyperparameters that you can use to stipulate or define your embedding. And the most commons are perplexity. And the perplexity is basically the radius of the t distribution that you use, how many cell neighbors you consider, either 30, 50, or so on. The number of iterations, as in the last slide, how many steps you do until you get to a nice conversion. The learning rate, and that I'm not much talking about today, and the theta, which is used for this optimization, and this speeds a lot the computation. Rather than computing the exact position of the cells, you compute an approximation of them, and that is quite fast. Which you can also, I recommend you to go to this link. This link has an interactive way of tuning those parameters as a tSNE. So you can see actually what exactly each one of them do. And they have many examples as well. So maybe after the course, and then you can go to there, or maybe tonight. But I'll have also some important notes about tSNE. Is that unlike PCA, tSNE is stochastic. So it's not like a computation. PCA will always look the same when you do the computations. For tSNE, you need to set a seed. So every time that you compute, you assign those points stochastically. So if you set seed, that stochastic will be always repeated. And the cost function never reaches a 0. Let's say that there is the defined, or there is the perfect tSNE to calculate. So it's up to you to define when to stop, or which plot is actually good or not to represent your data. So the cost function actually minimizes the local embedding. Now I'm going to start talking about local and global embedding. What do I mean with that? So for example, the distance between groups are slightly meaningful. And so for example, if you picture this cluster here, you have this pointy shape here, and you also have this pointy shape here. So in two different embeddings, this structure of the data is conserved between the two runs. However, the distance between this cluster and that one, so the furthest neighbors of that cluster, it's not actually meaningful, because tSNE is not optimized to find the distance between this one and the furthest one, but only within the local structure. The same thing goes for example, this cluster and that one, and they are also here. So this distance is meaningless in the tSNE, okay? This distance within this cluster, as long as they are kind of clumped together, this could also mean something, and you can even use it for trajectory analysis if needed. Yes, and the problem also with tSNE is that you cannot just add more samples. So if I have a data set and I have another data set that I want to combine, since every run of tSNE is stochastic, I just need to combine the data and run everything again. Yes?
Yeah, I've seen people doing clustering using the UMAP method and following tSNE.
That was a quite common strategy to use, but I would highly recommend going for a graph-based, because on the tSNE, you lose a lot of the resolution. So you compact your data to this two-dimensional space, and you lose some of the relationship between clusters and the cells. So it could be biased. If you have like a fast way approach to do that, because this density-based clustering that you probably maybe will hear today is quite fast, right? So you will be still OK if the clusters are quite well separated. Yes. Yes.
Yeah, here, like you said, that it's not, the distance is not meaningful, but it shows that they are separated anyway, right?
Yes. So we can be sure that those clusters are, maybe not be sure, but I mean, there is a good evidence that they are different, but how different they are, we cannot be sure. So even if they are closer here, it doesn't mean that those two are closer than this one. So that notion of distance is lost in this tSNE. Actually, to be more correct, for the tSNE, that also depends on the parameters that you use. So if you define the perplexity of the number of neighbors that you consider, you could, in theory, also get a global embedding. And there are actually papers tweaking the tSNE parameter, so then it actually can have this global structure of the data. But as a default, tSNE only do local embedding, or it's more specialized for global embedding. Yes?
Colors here, it's not made by tSNE, right?
No, the colors are 3 different samples. And I would expect them to be like this in, yeah. And this one is just kind of all together merged. It's just that it didn't have much iterations enough to separate it. So yeah, that's it. So there is the newer version of the tSNE, which is a fast Fourier transform accelerated interpolated based tSNE. I'm not going into details, but this is one of the versions that is already implemented in Surat, and there is also possibility to use it, and it's much, much faster. So just comparing to the previous one that is already quite good, Barnes-Hood's implementation or the Fourier transformation, and then you can embed 250,000 cells within an hour or even less. And I'll come to that in a little bit later comparing to UMAP. So a summary of tSNE. It's a non-linear graph-based method. It's so far it's still the current gold standard for single cell visualization, but now we're actually transitioning to UMAP. No, we're in that phase now. And can be run, for example, in the top principal components. So you can choose 10, 20, depends on the data set. But it has also some problems that it does not learn a specific function to put more points. You need just to run the whole thing again. The cost function is not convex, so there's no way to accurately say that this tSNE is close to what your sample is, or in a multidimensional space. And of course, there are many parameters to be optimized. You have 10 hyperparameters that you choose. Most of people only know 3 or 4. And in general, as by default, it does not preserve the global structure of the data, just the local neighborhood. So the distance between further clusters is not meaningful in tSNE. Any questions so far? No? It's going well? Yes?
It's not the clustering method by itself, right?
No, tSNE is just a dimensionality reduction method. And in some cases, for example, there are many methods for the density-based clustering. So you can cluster based on how the clusters look like here.
So it's just a visualization.
Yes, yes. So you'll hear more about clustering in the next session later, yes. So UMAP. So UMAP is a quite new method, and it's heavily implemented in the mathematical setting rather than only on the computational side. And UMAP stands for Uniform Manifold Approximation and Projection. UMAP is based on the topological structures, which is, I'm sure probably none of you is mathematician to understand that, neither am I. And I also had a hard time trying to understand this. So the idea here is that the points can be connected, or so for example, imagine that you have a point here. It could be 0, 1, or it could be the gene expression, gene 1, 2, 3, and 4, within your multidimensional space as for the other methods. And then you can connect those two. But now these, as a topological mathematical structure, these would be considered as simplex. So this is a way to represent this structure. So if you have a 2 simplex, let's say 3 points that are very close to each other, these form a topological structure. Topological, I mean like a surface. So if you have 1 point is 1 dimension, 2 dimensions, 3 dimensions, and then you can create structures, or a ball, let's say, or a square, or in this case, a thetahedron. And this thetahedron represents those 4 samples in a multidimensional space. I know it's kind of hard to visualize that, but here I'm trying to get you to the concepts that you can basically create a simplex out of your whole data. So this is, for example, these 4 samples are really well connected, and they probably represent the same cell type, maybe, and they are then connected to the next ones. And this is one way that you can calculate the distance of all your samples all together and get the structure of your data in this topological manner. And this way is how the UMAP can, instead of assigning the points randomly like tSNE to your reduced space, it actually calculates the distances based on this topological structure. So, and this is actually validated mathematically by the McInnes, and this is the the clever step that he has implemented or developed, in which that this speeds up a lot of the computation power that you need for the tSNE. Rather than trying to solve them stochastically, now you already assign which spots would be good for the embedding. So let's try to visualize that in another way. So this is also the example that he gave in his presentation, Mike Ines. So imagine that you have all those points, and then you can define an area where those points would be connected. So for example, those ones that they share a shaded line, so for example, like this, you'll connect this point to these other two because they share some parts in here. This dot will not be connected at all, neither this one. These ones will be connected to these two, and so on. So this is how you start building your simplex. So for example, here you have this three-dimensional structure that represents your data. This is very well connected until here that you have maybe just a triangle. So it's two simplex, one simplex, and so on. And the other trick that UMAP does is that rather than considering the Euclidean distance or the simple distance between points, the method is called UMAP uniform manifold approximation. Imagine that you have 2 points in here, this one and that one. And actually, they are separated in this real space, let's say like those two here. What it does, it applies a function that converts that those 2 points in here, they could be together if you transform that notion of distance in your dimensional space. So for example, the distance between those 2 points is exactly the same as the distance between those 2 points. It's just that the notion of distance in this part of your data is much more like elongated compared to this one. So if you draw, and because of that, you can actually have a function that you convert your real space to your uniform space. So yeah, so the distance now in this real space are actually variable, not just a single one as it would be for the tSNE. And now you can define, for example, every circle here, according to this transformation function, is the same as the other one. So for example, just because these ones are very well dense region in your map, these circles will be close together compared to these ones that are very sparse as data is concerned. And then the circle will be higher. But the radius of these circles do not matter. What matters is that the distance between this point and the length here is exactly the same as the distance between this one and that one. So that allows the UMAP to connect basically every single point in the data, or maybe not every single point, but the furthest points as well. It's quite complicated, I'd say as well. But then you also can have just like the tSNE, a fuzzy representation of your graph. So you don't really consider all the furthest apart, but at least at some degree. And then you can do that several times, and then now you connect the whole data set. So that's how UMAP would get the sense of the global structure of the data and also the local embedding. So coming back to the previous example that I gave for the tSNE, tSNE will care mostly about the distance between the red points or your local communities. UMAP now will consider also the distance between your closest cousin and also the other families in your community, in your data structure. And because of this feature, when you run UMAP several times, on maybe even in the same data set or parts of your data set, what you get is that the representation is exactly the same or very close together. So the clusters will always match together in the final embedding. So here you have in red and blue the 2 parts of a data set. So you have a data set that is split into 2 randomly, and then you try to do the UMAP on both of them. And then this is if you run UMAP, and this is if you run tSNE. And the tSNE doesn't really matter because the positions are defined stochastically. And because of this feature as well and those mathematical tricks, so then, for example, the average distance between the points, so for example, the distance one cell here and then the corresponding cell in the red data set will be kind of here. So the distances in the UMAP, if you run them twice or 2 different data sets and try to compare them, will be very low compared to the other ones, to the tSNE, for example, or large V's, which is another graph-based dimensionality reduction. Yeah, and this is also correlation of mapping position. So UMAP is quite good to assign new points to your embedding in that way. So you don't really need to run UMAP if you have more samples. It's still recommended to do that, but it's just another way that you can do without it even. Yes?
Do we care about tSNE anymore?
That's a good trick that I'm not sure yet if we are ready to discard tSNE, but UMAP is really good. Many researchers still prefer to have, for example, if you have clusters, them separated, and just for cluster or visualization of your data. But I think we are going towards more UMAP. And since we have and I'll come actually to that, to the Fourier transformation of the tSNE, which is as fast as UMAP. So it doesn't really matter which one you choose, because yeah, in many cases you'll get kind of similar results, especially if you tune the tSNE to have also the global embedding. So they will be quite similar in the end as well. Yeah. So the UMAP assumes that there is a structure in the data. And depending on the tuning parameters, and this is the same thing for the tSNE, you have 10 tuning parameters that you can choose. And you can generate artificial clusters based on those definitions. So there is not one parameter suits every data set. Every data set needs to be tuned a little bit to fit your data. And how you evaluate that? Using biology. So you need to know or have, for example, differential expressed genes between that cluster and the other one. Otherwise, if you generate two clusters and there is not a single differentially expressed gene, then that is a good indication that these are clusters that were generated artificially because of your method. So they should actually be together. Or maybe a data integration issue. So this is a data set. So this would be like the default data set that is using all of those plots in here, but then changing the number of neighbors from 5 to 200, 300. And here also the minimum distance between points. You can also see that you start seeing some kind of clustering within that data set, even though the original data is not, there's no cluster at all. It's just a uniformly distributed sample. And I also highly recommend to read the documentation on UMAP. There is also quite good documentation on that and explaining how to tune every single parameter. So comparing most of the current methods, UMAP is quite fast. And also the Fourier transformation of tSNE, as you can see here in purple and UMAP in blue, you can see that they are quite scalable up to millions of cells. So either of those will be OK to run. And this is how the UMAP representation seems like. And you also have used it yesterday. So you have an idea already how it looks. But now, what is the difference between this UMAP and the tSNE? What can I assume, for example, from here? Is the distance between this cluster and that cluster relevant? Yes or no? Or I don't know. I'm freaked out. Yes. So 1 yes. So 2 yeses, 3 yeses, 4 yeses. Yes. Who takes more? Yeah. So this distance now matters because UMAP got the global embedding, the global structure of the data. So to keep in mind, UMAP is a nonlinear graph-based dimensionality reduction algorithm. It's very efficient, as well as Fourier transform tSNE. It can also run on the top principal components, which is actually what you're going to do. And other things that I haven't mentioned much. Usually you use Euclidean distance, but actually you can use any distance. So if you have apples, pears, and oranges, as long as you know how you can measure the distance between an apple and a pear and an orange, you can use that matrix also in the UMAP. So it's generalizable in that sense. And you can also integrate between different data types. So oranges, pears, plus gene expression, plus if it has a car or if the pear doesn't have a car. And it's also no longer stochastic as the tSNE and defines both local and global embedding of your structure, of your data. And also you can apply it to new data points because the embedding is quite similar between the two different runs. So as long as you have enough samples, the embedding should be very similar together as well. And considering that the data sets are also quite close, let's say. So if you have bone marrow and you put another bone marrow, and then you can map them together. So as a wrap up, here are different single cell workflows. So as you can see, this is updated from January, I think. the last time I gave this presentation. So Seurat version 3, Scatter, Pagoda, and Monocle. So you can see that all of them have PCA, all of them has tSNE, and nearly all of them has UMAP. So regardless of the analysis pipeline you choose, you always have those methods because they are quite standard. And there are different ones for the different one. For Seurat, for example, they have also diffusion maps, large Vs for Pagoda, and large Vs will be also quite similar to UMAP as well. Yeah, and there are other dimensionality reduction algorithms. Maybe I'm also missing some of them here. And there, I will highly recommend also to read this other paper that compares some differentially, some dimensionality reduction algorithms. Yes, that's it. Do you have any questions so far?